Hello, everyone. Thank you so much for joining us today. Welcome to the April session of the Research Seminar Series. My name is Cheyenne Head, and I'm a research assistant with the BC Brain Wellness Program, and I'm very excited to be hosting today's session. We have Dr. Shannon Kolind and her research team speaking to us today, who will be talking about novel imaging techniques in brain health research. Before we dive into the session, I would first like to acknowledge that the BC Brain Wellness Program at the Javid Moafagian Center for Brain Health is situated on the traditional, ancestral, and unceded territory of the Hunkaminam-speaking Musqueam peoples. I am also joining from Vancouver and would like to invite you all to take a moment to acknowledge and give thanks to the land you are joining us from today. There are a few housekeeping things before we get started. If you have any questions or thoughts throughout the event, please feel free to send them in the chat box and we'll get to them during our Q&A session. Please note that we will also be recording today's event and uploading it on our YouTube channel. Today, I'm very excited to welcome Dr. Kolind, Adam, Poljanka, and Sharada. They will be presenting on novel imaging techniques and how their team is developing new methods to image myelin in the brain and spinal cord. This research team is focused on developing a toolbox of tissue-specific imaging techniques and making them available to everyone, everywhere. Without further ado, I will be passing it on to Dr. Kolind and her team.
Great, thanks so much. I just wanted to start by thanking the organizers all for having us speak today. So as introduced, my name is Shannon Kolind. I'm an associate professor based in neurology, but I'm a physicist by training. So what our lab does is we develop new ways of using MRI and then apply them to look at brain health. And that includes looking at neurological disease. In particular, we tend to look at multiple sclerosis. So MRI uses magnets to look inside the body. And 20 years later, I'm still absolutely amazed by that. I think it's so cool. So unlike X-ray, which is really good at seeing bone, MRI is actually looking at signal from water. And luckily, we have a lot of that in the brain. So we're quite good at looking in the brain with MRI. So I'm really lucky to get to work with these three amazing PhD students who are going to share some of their work with you today. And we're coming from across a kind of a broad range of backgrounds representing neuroscience, physics, and engineering. And they're going to tell you about how we can use MRI to give us more specific information about biology in the brain than conventional MRI does, why that's important for brain health, and how we're trying to make that technology available to everybody. So I'm going to start by passing that over to Poljanka Johnson. This one. Take it away, Poljanka.
All right, thank you so much for that introduction. So before we get started, we have to define what is myelin. So electrical signals need to move around our brain and spinal cord to tell us what to do, what to say, and everything in between. These electrical signals travel along our neurons. However, our neurons tend to be quite long, so the signals need some help to get down to the end of the cell. Myelin is a fatty tissue that wraps around the neurons. You can see it in this first image here on the left, and insulates the neuron to allow the electric signal to travel faster and further, kind of like an insulation on a wire. So without myelin, signaling in the brain becomes a lot slower or non-existent, meaning that myelin is a very important metric for brain health. So we can actually image myelin, which is the big technique that we'll be talking about today. So here at UBC, we've developed a technique called Myelin Water Imaging, where we can quantifiably measure the amount of myelin present in a region of interest, whether that be the brain or the spinal cord. So on the left, that nice blue image is actually just a picture of the Myelin Water Imaging. And you can actually see some structure you can pick out. It looks like a brain. It does look a little grainy, however, but that's because each pixel in this image has a value termed the myelin water fraction, which is what we use to measure myelin content. So the image on the right, which is the one with the little peaks in the blue and purple, is an infographic on how we measure the myelin water fraction. So the MRI machine uses a radio frequency pulse, which is nice because there's no ionizing radiation, which means no cancer. To excite the water in the brain, as Dr. Kolind mentioned, we have a lot of water in the brain. The water exists in various layers. So there's water, or various regions. So there's water between the layers of myelin, and then there's water inside and outside of the cell. So once this water gets excited or more energetic because of the radiofrequency pulse from the MRI, it will then lose this excitation. The water trapped between the layers loses it far more quickly than the intra-extracellular water. So you can actually see Myelin water loses it between 0 and 40 milliseconds, and then intra-extracellular water is 40 to 200 milliseconds, which is a very big difference. And we can measure that. And so the way we calculate the Myelin water fraction is just the fraction of the signal from myelin over the whole thing, hence myelin water fraction. And this does actually correlate to the amount of myelin present. And we have shown that, and we can give you references. But next, there we go. So before we get into the diseases, we're going to cover the healthy brain. Your myelination does change throughout your life. So you are born with relatively low myelin, and you go through a stage of rapid increase until about year 3, and then it steadies out a little bit more until adolescence. And then it actually keeps increasing, albeit a little slower, until about the age of 50. And people have studied myelination in adolescence and childhood, and they found that disrupted, altered, or less effective connections between brain regions, especially supporting academic learning, leads to or is related to learning disabilities, such as dyslexia. So there was a paper that showed lower myelin water fraction is correlated with dyslexia. So as we get older, now we'll go into aging, our myelination tends to follow a U-shaped pattern. So we get more and more and more, and then we get less and less and less. And different regions of the brain myelinate at different times. However, the later a region of the brain tends to myelinate, the earlier it tends to demyelinate. So this is a last in, first out hypothesis, and you can see this with the image on the right here, where it does follow that U-shape. It is important to note that you can actually slightly modify this amount of myelin that you have with healthy habits. So exercise, mental and cognitive exercise, so keep thinking, will actually increase your amount of myelin, which is very important for aging. And if you do develop these demyelinating diseases that we will talk about in the next slide, because you have brain reserve. So it'll take longer if you have more myelin to show these demyelinating effects clinically, which is great. All right, so demyelinating diseases. I'm going to cover 2. There are quite a bit more than 2, but these I thought were probably the biggest and most relatable diseases. So the first one is Alzheimer's disease, which everybody I'm sure has heard of from memory loss, and it is a truly tragic disease. But before people tend to develop the hallmark symptoms, people have looked at early Alzheimer's and genetics. So there is an ApoE4 gene, it's a lipoprotein E4. It's naturally occurring within the general population, and it is a gene associated with risk of Alzheimer's disease. It increases your risk by about 1.5%. So it's not that big, but it is there. And just having this ApoE4 gene is associated with reduced myelin water fraction. So there was another study that looked at cognitively normal. This is the image on the top right. Cognitively normal, mildly cognitively impaired, and Alzheimer's disease. And they saw that only in those people with Alzheimer's disease had a reduced myelin water fraction. So even if you had mildly cognitively impaired, you didn't show this reduction. And that myelin water fraction loss in Alzheimer's disease was correlated with memory loss. So next we'll move on to multiple sclerosis, which is hallmarked, at least on MRI, by these demyelinating plaques. So you can see these little holes. I don't know if you can see my mouse, but there's little like holes essentially in the brain, and that's regions of loss of myelin. And so yes, demyelination is visible in lesions with MS. However, you also have normal appearing white matter. So it looks regular on conventional scans. But with Myelin Water Imaging, we have shown that even normal appearing white matter has lower myelin water fraction. And even further, this decrease in myelin water fraction is correlated with clinical measures of MS. So there was a study that came out of our lab, actually, about a year ago. I'm going to plug it because I'm proud. Where they looked at the myelin water fraction, and then they collected clinical measures of MS. So measures of disability, cognitive scores, and a battery of tests, and they found that there was a link between the myelin water fraction and normal appearing white matter, so not even lesions, and that clinical score. All right, moving on to one last thing that is very cool, traumatic injury. So this is a pilot study. They only had 3 human participants, but their results were really cool, so I thought I'd share. These 3 people had a partial injury of the spinal cord and were undergoing rehabilitation, which is this kind of erratic downhill walk. And so after they completed the rehabilitation, they saw up to a 40% increase in myelin in brain motor learning regions, which is massive. That is like a massive increase. And so just to double check, they mimicked the same injury in mice and had them go undergo this rehabilitation. And they tried to see if there was actually an increase in myelin, and they saw not only an increase in overall myelin, but an increase in the new myelin. So there were new cells that were creating new myelin. So this just highlights how versatile and important myelin water imaging is, and just how much we can use it. And with that, I will pass it off to Adam.
All right, thanks, Poljanka. So Poljanka's given us a pretty comprehensive overview, fairly convincing, showing that myelin water imaging is quite a powerful tool for understanding the healthy nervous system and also diseases. So I'm Adam, I'm a physics PhD candidate, and I'm going to tell you about a bit of the research that I've been working on also in Dr. Kolind's lab, more on the technical side of how we can improve myelin imaging itself. So the first thing I'm going to talk about is how we can better understand myelin imaging results. It's really essential when you have any type of MRI to be able to understand if something's abnormal. You have to have a really good understanding of what normal looks like. So here on this slide, you can see a bunch of different structural, conventional brain MRI scans. So these are showing the different brain regions, and this is not myelin imaging, but even just with these types of images, you can see how diverse different brains are. So I kind of like to say that brains are a little bit like a fingerprint. Everyone's are different sizes, different shapes, they have different characteristic patterns, and you can see all these little folds in the outer edges of the brain, and that makes it really difficult to compare different brain images, because to do that, you have to be able to line them up with each other somehow. That's necessary to make sure that you're actually comparing apples to apples and oranges to oranges, and not totally different brain regions. So one of the first projects I worked on was to try to tackle exactly that problem. I worked with a technique for registering different images together. So you take a couple images, and then sort of like if they were made of clay, you can mold and massage them, and then you can line them up together. So I actually collected data from various studies that have been ran here at the Center for Brain Health from 100 different healthy volunteers. And these were from people without brain or spinal cord injury, and they span a really, really wide range of ages. Now that's really important because as Poljanka mentioned, your myelin content will actually change throughout the lifespan. So what's normal for someone who's about 20 is different from someone who's about 50, and different from someone who's about 80, and so on. So we had a range of people between about 20 years of age to 78, these 100 individuals, and each of them had what's labeled in here as a 3D-T1, and that you can sort of think of as like a normal MRI scan. So that's showing the structure of the brain. And then we have these other scans, which we have labeled as grays, which is this specific technique we used, but that's actually the data that we collect for myelin imaging. So we're showing one brain image here, but actually for myelin imaging, we acquire about 50 separate images in the same acquisition. And we use those 50 different images to calculate what Poljanka was describing, the different water signals that are coming from within the brain. So overall, we have these structural images, and they're three-dimensional maps of the whole brain. And then we also end up with three-dimensional maps of myelin from within the brain. So once we've merged these all together, we can create what we call an atlas. So on the left, we have sort of a structural atlas, which they also call a template sometimes. And that's the structural images for 100 different healthy volunteers all merged together. So this creates sort of a representative average. And I've actually had in the past, I show as an example, we can even use the same image registration techniques and take headshot photos from all our lab members, and we can register those together and create a sort of representative researcher from our group. So this is, they're really fascinating techniques that are actually not specific to MRI, but they work very well here. So we have these, this structural template on the left, and then the second column from the left in blue and green, that's our myelin atlas. So that's showing the distribution of myelin throughout the brain. And that's actually, as I mentioned, a three-dimensional kind of roadmap that you can scroll through, and you can look at different brain regions to study differences in myelination. And then on the right, we also have these maps that are in sort of a yellowish red color scheme. And that's because, as I mentioned, we have 50 different images from Myelin Imaging. That's a lot of data. And there's actually quite a few other things that we can do as well. So we mentioned we can study myelin by studying the fast relaxing myelin water. We can actually also study the other water compartments. And that can tell us quite a lot as well, not so much about myelin, but about factors like inflammation and iron content and water content, which are all things that are very often affected by different diseases. So that's what's being shown in the right two columns there. So once we have this atlas, you see we have this brain atlas on the left there that shows the distribution of myelin. And we've actually done the same thing in spinal cord. So spinal cord is also part of your central nervous system, and it sort of relays the signals that are generated in your brain throughout to the rest of your body, to your peripheral nervous system. And so on the right, we have a myelin imaging atlas for spinal cord as well. You can see that sort of butterfly shape in the center. That's gray matter structure. So we were able to get a fair bit of detail in the spinal cord, which is only about the size of your pinky. It's very, very tiny. And part of that is from these really effective image registration techniques. So once we have this atlas, what is this useful for? Well, sort of mirroring what Poljanka was talking about, we can use the atlas to study healthy brain. So as I mentioned, it was created from 100 volunteers without brain or spinal cord disease or injury. And so we can actually look at a kind of overwhelming, terrible looking graph here, but all these are each of these little plots is a different region of the brain. But what we're seeing from left to right is different age groups. So on the left, we have people grouped from ages 20 to 29, and all the way on the right is 70 to 79. And so this is, again, what Poljanka was showing earlier on, something that we can really characterize quite well using this atlas is how myelin content changes with age. And also, as Poljanka hinted to, we can study other demographic factors, a lot of which are quite interesting, things like years of education, although it doesn't necessarily have to be in school. Anything you do keeping your brain active is going to be beneficial. And even things like whether you're left or right-handed will affect asymmetry of myelin values. This is just another plot that's showing the same thing, but plotted in a different way. And the sort of key takeaway here is that we're looking at all these different brain regions, and we have those grouped age groups, and we can see the trend across ages is really consistent between all these different brain regions. And another interesting thing is that we've gone this whole MRI route to study myelin and how that changes throughout the lifespan, but there are actually post-mortem studies which have been done on brains donated by people at various ages, and it agrees really closely. So it's very cool to see that we can use a very safe, non-invasive technique to actually probe the same kind of tissue parameters. So we've talked about the Myelin Atlas and then what we can learn about healthy tissue. So now I thought I'd mention how we can apply something like the Myelin Atlas to better understand diseases. So Poljanka mentioned a few different diseases, but one of the issues when we're studying disease, especially diseases that cause structural changes to the brain, is that I mentioned brains are all quite different, and brains that are affected by disease are often even more different. So on the left, we can see a structural MRI image from a person living with primary progressive multiple sclerosis. So this is a sort of more aggressive form of multiple sclerosis. And you can see the shape of their brain is very different than our representative average template on the right. Luckily, we can use those same image registrations that we use to create the atlas, and we can align the images with each other. So we can put the patient brain lined up with the template, or we can line the template up with the patient brain. So that's really beneficial, as I mentioned, to be sure that you're actually comparing the same thing, especially when you're using these advanced quantitative MRI methods such as Myelin imaging. So once we've done that, the power of that sort of technique is that we can evaluate Myelin imaging on an individual basis. So traditionally, people would have to collect a huge amount of data from big groups of control groups, healthy volunteers, and then also groups of people with diseases, and then do statistics on that to be able to pull out results. But what the Atlas lets us do is once we've lined data up, we can see areas that we can highlight where there's statistically significantly low myelin values. And so those are highlighted on the structural image with these little flared red and yellow blobs that you can see there. So on the left, we have someone with a less aggressive form of MS. They have a low level of disability and they haven't had the disease for very long. And you can see their brain doesn't show a lot of evidence of pathology. There are a couple little spots. But we can also look at the image actually from the same patient that I showed previously with that registration example. So they have a high level of disability and they've had multiple sclerosis for almost 30 years. And we can see widespread regions. So both the extent and the magnitude, the amount that their myelin content is lower by is fairly extreme. As I mentioned, we can also do this in spinal cord. So again, we see the same story. This is the spinal cord data from the same individuals. You can see not really extensive changes in this relapsing remitting MS patient. And then the, oops, jumped too soon. The primary progressive patient, we see the same sort of thing. So on the left, we're looking at kind of slices through the spinal cord from the MRI. And then on the right, that's actually sort of a transparent 3D display where we can see blobs that are highlighted. And when you look at structural MRI, those blobs often correspond to lesions, which would often appear as bright spots or dark spots on traditional MRI. But the advantage with myelin imaging is then we can see if those lesions actually have demyelination occurring and by how much. And then something potentially even more important is as we move forward, trying to develop therapies for especially these more aggressive types of MS that promote remyelination, then myelin imaging is extremely valuable to assess whether or not and how much that's actually occurring. So the last thing I'm going to talk about is the actual myelin imaging itself. So the way that we acquire it. Shannon sort of mentioned this early on, but MRI is a really cool technology to work with because it's less hardware dominated than other types of imaging. And what I mean by that is that the hardware, the physical parts of the scanner are sort of limited by safety limitations related to putting a human in there. And so the main ways that we can improve myelin imaging or other types of MRI are by programming new ways to do it. And so it's kind of like all these MRIs all over the world are smartphones, and we can develop apps. And if they're working well enough, then we can share those apps with scanners all over the world. So this has been a really cool area to work in as well. And here I'm just showing on the left, we have sort of a normal MRI. So this is one of those 3D T1 structural images. So this is just one image. And then in the middle, we have our conventional approach for myelin imaging. And so this is one of the 50 images you acquire, but as you might imagine, acquiring 50 images takes a very long time. So to try to speed it up, we acquire it quite low resolution. You can see the image is kind of grainy and a much smaller field of view. So with Myelin imaging, we're just covering sort of the minimum amount of the brain there. This is actually already drastically improved, this technique I'm showing in the middle, because originally Myelin imaging would acquire just a single two-dimensional slice. So you don't have a whole brain that you can scroll through, you get one slice of data, and that would take 26 minutes long. And for people who've been in an MRI scanner, they know it's fairly difficult to sit still for 26 minutes. And if you move by too much during that time, well, then the data is not very useful. And so that's a lot of time, money, and effort wasted. So our goal is to make Myelin imaging faster, higher resolution, more robust, and ultimately more similar in quality to the conventional images like you see on the left. So that's actually exactly what we've been able to do using a technique called compressed sensing. So this is sort of a general mathematical technique that we've stolen from the field of image compression. So if you've ever had JPEGs or PNG images on your computer, they're stored using math related to compressed sensing. And actually astronomers use it quite often looking at stars because they have, interestingly, a lot of the same issues we do with MRI. They need to improve their imaging. So on the right there, we can acquire those 50 Myelin images at higher resolution and with a much larger field of view. So you can see it's covering all the way down to the spinal cord. That with normal techniques would take almost 2 hours, and we can do it in less than 10 minutes. That's awesome because it means that all the data we're acquiring for all these studies and all of the conclusions we're trying to draw about healthy disease and brains in general, healthy brains and disease as well, all of that data is higher quality. So it's much higher resolution. It has better repeatability, which means that if we scan someone multiple times and even on different scanners, we want those values to match up. It's kind of like if you have a measuring tape, you want it to be the same length all the time, and you don't want it to get wet and shrink if you measure something outside. That's really not ideal. So that's a huge advantage. And lastly, I mentioned that large field of view, which just means we're actually covering all the way down to spinal cord with one single scan. Lastly, I'll just show one more slide, which is an extension of that compressed sensing technique where we're trying to do it even better. So we're acquiring data at much higher resolution than we ever have for Myelin imaging, and also very, very quickly. So on the top, you see a scan that's well over an hour and a half long, and that's already accelerated, sped up with normal techniques. And then this caliper data set on the bottom, you see 2 of the example Myelin images, and then on the right, the Myelin water fraction. So that's the kind of Myelin map. And we can acquire this at resolution that's actually quite similar to normal structural MRI, but again, in under 10 minutes. So that's sort of a whirlwind highlight of some of the research we've been working on. And I'll now hand over to Sharada.
Hi. Okay, so we've talked a lot about what myelin is and its role in the nervous system, and Myelin water imaging and how it quantifies myelin, and then how we've made Myelin water imaging faster. So we're going to sort of switch gears a little bit and talk about how to make MRI more accessible as a whole and how to make Myelin imaging more accessible. So this is a map that I found about the Canadian Medical Imaging Inventory from 2019-2020, and it shows the distribution of MRI scanners across Canada. So if you look at BC alone, you see that most scanners are located around the Lower Mainland area. So this means that people who live further away are going to have to travel pretty far to be able to get access to a scanner. BC has 46 clinical MRI scanners, and these are all shown on this map here. And this also means that people have quite long wait times to be able to get access to scanners. So improving access to a scanner is actually a worthy cause. So this is a pretty conventional scanner with a person next to it for scale. It's pretty big. It's a pretty hefty investment. If you don't know what a scanner is, like Shannon was saying earlier, it's basically a giant, very strong magnet. So magnetic field strength in these scanners is measured in units of Teslas, and the scanner in this image a minute. And like conventional scanners and are often 1.5 or 3 Teslas strong. So this requires a lot of infrastructure because it's a super strong magnet. So it needs, for example, like specially shielded rooms. You can't have metal in there that might be attracted to the magnet. It needs really highly trained operators who have to follow a lot of safety protocols. It needs liquid helium to be able to cool parts of the magnet. So on the whole, it's like a really big investment and like a lot to handle. So the picture on the right is also an MRI scanner, but it's a lot smaller and a lot cuter. It's made by a company called Hyperfine. And this was the day we got it, just sitting outside the brain building where we are right now. And so the field strength I said of the scanner on the left was 3 Teslas. So the field strength of the scanner on the right is 0.064 Teslas. So that's like 50 times weaker, which is quite a lot. It's actually not a lot stronger than a fridge magnet. And it runs off of just wall power, and it's got wheels, so it can actually be driven around. And so this means that it doesn't really need any special infrastructure. And actually, we are sitting in the same room as the scanner behind us. So we'll do a little bit of a show and tell. Let me stop share. Okay, so the stretcher is not part of the scanner, but hopefully you can see all of this. So we'll start from the top. So this is called a 5 Gauss line, which is sort of a safety feature beyond which you shouldn't be taking laptops. And I will add, we're sitting around with like 5 laptops in this room, which is kind of amazing, and like 4 people. Okay, moving further down into the scanner, this is the space where the magnetic field strength is that 0.064 Tesla I was talking about. It's got magnets embedded into the top and bottom over here. So this thing is called a head coil, and it's this like clear coil. And this is the thing that like transmits and receives signals from a person's head. And this is where a subject's head would usually go. Oh. Smooth. Okay, so you can see that it's like a lot more, it's a lot less claustrophobic than a big scanner, which is one of the big pluses, and it's also a lot quieter. These things around here are called the RF cage, and they sort of take care of electromagnetic interference, but that's kind of the only shielding that we need for the entire scanner. Okay, moving further down on it. Oh, thank you. So this is where the computer of the scanner sits, and this is what tells the scanner what to do, what to run, and generates the images. And finally, at the very bottom here are the wheels, which are the other big magic piece, which is what makes it so portable. And all it does is it's controlled by this joystick. By this joystick over here. Okay. Okay. On the whole, it's not very big and it's fairly light. I think it comes in at about 1,400 kilos, pounds. Okay, hopefully we're back to this. And there's a little recording of how it gets driven around. So you can literally just unplug it and walk around with the scanner, which means it's great for within hospital use, for example, for taking it to patient bedsides rather than wheeling the patient into a big MRI scanner. And it's also good for example, sticking in a van and driving it to remote communities. So it's really solid for accessibility. Here are some example images from the scanner. So this was some fruit that we scanned on, I think, one of the very first few days that we got the scanner. And this leafy thing in the middle is an endive, which is fairly small. So you can see the separate leaves, which means it's like pretty good resolution. This is an example of a healthy volunteer's brain. And again, you can see pretty fine details. So this is kind of amazing for something that comes from the strength of basically a fridge magnet. And I think the total cost of it comes in at under $100,000. So it's also very cheap as far as scanners go. So let's look at a couple of use cases of it that other people have done. So this was something that came out of a group at Yale University, where they applied the portable scanner, the Hyperfine scanner, to intracerebral hemorrhage. So in this picture here at the bottom, in the rightmost column, which says conventional, that picture came from a 3 Tesla scan, so a pretty standard clinical scanner. You can see the red arrow pointing out this white region and then this dark patch. These PMRI images are portable MRI images, and these are 2 types of images acquired on the portable scanner, like the one behind us. And again, you can see the same pathology shown up in the portable scanner. A different example this time, this is a cerebral infarct, again from the same group at Yale. Here, the rightmost column is SOC, which is standard of care, and this is a CT image of the infarct. You can see this like shaded region on the right hemisphere. These POC images are point of care images, which is from the Hyperfine scanner, again, like the one behind us. And you can see that same region sort of highlighted in each of these different types of images. different example this time. This is a group at the University of Pennsylvania that applied this portable scanner to multiple sclerosis. And multiple sclerosis has a characteristic feature of lesions, like Poljanka was talking about, which are regions of demyelination. And like Adam mentioned earlier, lesions show up on scans as like bright or dark regions. So for example, on this 3T scan, which is the one on the right, so these like really bright white spots, and these are lesions. And they wanted to see if these could be seen at 64 millitesla as well, and they were able to. We actually did pretty similar work to this, where we acquired similar images on some postmortem brain with lesions as well, and our findings lined up pretty well with what they found. So that's pretty encouraging for repeatability too. Okay, so it's pretty cool. Lots has happened already, but what are we trying to do with it, and how does this tie into myelin imaging? So we hope to be able to implement techniques like ours, like Myelin Water Imaging, at low field, so that we can look at pathologies like multiple sclerosis, for example. And we're also part of a study that looks at early childhood brain development in lower and middle income countries. So a lot of these countries have different sites which already have hyperfine scanners. So we intend to develop these techniques and then distribute them to these other sites so they can go into use pretty much immediately. So really the hyperfine is opening up a whole new avenue of research and accessibility and like new applications. So it's a pretty exciting time to be an MRI. And back to Shannon.
Great, awesome. All right, so we just wanted to wrap up. So before kind of getting to conclusions, I just wanted to make sure that we said thank you to all of our volunteers. We're so lucky to have so many people that are so generous with their time coming in and jumping in the scanner for us. Our MRI technologists who run our 3 Tesla scanner very expertly, and all of our sources of funding we've highlighted, I think hopefully all of them are on the sides of the slide there. And I just wanted to talk very quickly about some of the ongoing studies that we're doing. So a lot of these tie into what you've heard about from the students. So we have this ongoing study, Poljanka highlighted, where we're looking at myelination in MS on our 3T scanner. And so we already showed that the myelin measurements correlate with things like cognitive disability. And we're now following that up over the longer term to see if we're able to predict longer term changes in MS with that snapshot. And this is really useful for clinical trials. And actually, we are using these techniques in clinical trials now for particularly MS therapies, but we're expanding actually to quite a few other applications such as Alzheimer's and Parkinson's disease. And it's turning out that there's a bunch of drugs coming to market aimed at remyelination. So this is a really useful technique for something like that. So we're helping out with some of those as well. We're also looking, as Sharada mentioned, at comparing what we can see on this little 64 millitesla scanner, the baby scanner, compared to the daddy scanner for MS, see how this can be used in diagnosis and also myelin assessment. So that's ongoing right now. We have a big national study called CanPro-Co that some of you might have heard of, and this is across 5 sites across Canada, where we're looking at 1,000 people living with MS over at least 5 years. So this has been really exciting to try to harmonize these studies across all these different types of scanners and different centers. And we'll get just a ton of information, including blood biomarkers, MRI, and health outcome measures. And then Sharada also mentioned Unity, which is this project that is funded through the Gates Foundation, where we're looking at neurodevelopment in youth, particularly in countries across sub-Saharan Africa, as well as India and Pakistan. So as part of this study, 10 scanners have already been shipped to those locations, and they're starting to use them in real world situations. And we are getting partnered with those sites, and we're developing in real time techniques that are useful for the things that they're trying to study in these kids in particular, which has been just absolutely inspiring. And hopefully this year we'll get to go visit some of those sites in person and really get to see how we can really make an impact. Adam talked about the Myelin Atlas, and we're continuing to build that all the time. So anybody who's doing a study here, we invite them to contribute data to that atlas so we can make it bigger and more diverse, and so we can learn things about the healthy brain as well as use it as a more dynamic basis, as you talked about comparing individuals too. So future directions, and we'd love to hear impact from the audience on where we should go with all this technology. So this was the first of these portable MRI scanners in Canada. There are still only 3. This was the 38th one built. So this is a really new technology, and we're still trying to figure out how this fits into the landscape of healthcare and research. So we'd love to hear your thoughts about if you think this is exciting and where you think it could be best used, how it might benefit people. And we're also, of course, working on expanding the Myelin imaging across all platforms, as Adam talked about, making this work like an app that we can distribute across cell phones as well. We are always looking for volunteers, so please feel free to drop me an email if you're interested in hopping in one of our scanners or just feeding back on some of our studies. We always really love to hear from the community. And we'll stop there and open for questions. So thank you very much.
Thank you so much to all four of you. That was really wonderful and very stimulating. It's so fascinating to see how far MRI technology has come, and it's incredible to see how efficient and condensed that portable scanner is. So yes, it's definitely an exciting time in the field and some exciting ongoing research as well. We do have a few questions here from the chat. Can you give us an idea of degree or measurability of improvement in myelination with exercise, and how much exercise? And I guess a kind of related question would be how we increase myelination, and is myelination always better? Is more myelination better?
Yeah, great questions. So this Myelin Imaging, water imaging, was pioneered here at UBC by Alex McKay, who you actually saw standing next to the 3 Tesla scanner in Sharada's picture. So we're super proud of that technique, but it's really only in the last about 5 years that it's become accessible and usable at multiple centers. And it has been taking off really quickly, but as a result, the number of studies that have been done with it are somewhat limited, but there have been studies with it showing that in as few as 10 sessions of playing a video game, we can see a difference in myelination. And in terms of physical exercise, definitely there's been differences shown in people who just do like a regular 30 minutes 5 times a week compared to people who are more sedentary, that you can see overall just a bit of a shift in the level. But it is individual to different people, much like we all have different heights and fingerprints and that sort of thing. But it is something that you can change quite rapidly and do throughout your life. And essentially anything that you do that's different is going to start myelinating a new pathway. And if you do it for enough times, it's going to stay myelinated. So your brain's going to say, okay, this is important. I'm going to cement this in. And there's a famous study out of London where the taxi drivers have one of their brain regions is extra big from all of memorizing all of these different directions and routes and having to change course as things shift. Of course, all before we had GPS in our pocket. So basically anything that you can do that's different, get out of the regular routine, playing like crossword puzzles, interacting with new people, anything different is going to help with building up that myelin.
Wonderful. That's exciting. It's good to know. And can myelin imaging be used to detect Alzheimer's disease particularly for early diagnosis of Alzheimer's?
Yeah. Do you want to talk about that a little bit? Or
You can start.
Okay, sure. Yeah. So again, this is kind of a relatively new area, but we definitely think this is an exciting avenue. So there's a few studies that are starting to look at this. So there's some work showing that you can already detect changes in mild cognitive impairment, as Poljanka was talking about. So kind of before it becomes Alzheimer's, but where it might be leading up to it, we're already starting to see some changes. And so that might be an opportunity for intervention. So if we can prescribe various exercise, whether it's physical or mental, or potentially remyelinating therapies and this sort of thing. There's also a huge study called the Canadian Longitudinal Study on Aging. And so this has been looking for quite a long time at measures of aging, and they have been adding MRI components and just more recently Myelin imaging components. And this got added actually as a result of COVID-19, one of the very small number of good things that hopefully come out of this horrible couple of years, where they were able to get some funding to start looking at the MRI in cases of people who have or have not contracted COVID and how that's affecting aging. And so we're able to now expand on that and include it in all of the studies going forward with the aging study. So we'll be able to really see this in a larger group. And so then we can look back and look for some of those early warning signs. But essentially by building up things like the Atlas, we hope that this will be actually useful in the real world in the next 5 years.
So I guess given that it's not regularly used in clinical care right now.
No, it's not. We are seeing it used more and more in clinical trials, but not kind of in routine clinical care. At UBC, we have a really good relationship with a lot of the neurologists, and so they've taken a real special interest, and it's been really great having that interaction. So for us that tend to be more on the technical side, to see where it can actually be useful in clinical care in these cases, and for them to see that there are tools outside of kind of the regular toolbox. And so, for example, there was a case a few years ago of a young lady who had radiation for a potential tumor, and then they had some unexpected changes. And so we took some of our advanced MRI techniques, and we just, kind of threw all of the new techniques that we could to look at this thing, and we were able to find out a lot more about it and actually kind of guide the treatment. So that was really exciting in that we could participate in active care, and that she did seem to improve as a result of finding out more of that information. So it's definitely on the cusp.
That's great. That's really exciting. So do you see in the future that this will maybe have a role in MS assessments in clinical care?
Yeah, absolutely. We really hope so. So the conventional MRI has been absolutely revolutionary for MS, and it's one of the reasons that it's one of the only neurological diseases that does have effective approved therapies. It's not a cure, but it's a lot better than nothing. And that's because of being able to see those lesions on the conventional images. But with the Myelin imaging, we can see not only those lesions, but also those changes in the normal appearing tissue. And so there's kind of these two aspects to MS. So there's the relapsing remitting component where you have an attack, and then you have a period where things are fairly quiet or maybe even improving, and then another attack, and then you have this progression. And so some people are diagnosed with progression right from the start, where they're just kind of steadily accumulating disability. At others, it might be a little bit more quiet, more in the background to that relapsing remitting, but we do think it's probably always there to some extent. And so being able, it does look from the studies that Poljanka was talking about, like is the changes in that diffuse myelin is more linked to that more gradual progression. So being able to detect that in a small period means that we can intervene sooner, and especially in a healthcare system that doesn't approve some of the more aggressive drugs until you can show that there's damage. Wouldn't it be nice to be able to show that you're maybe going down that path before you get there and do something about it earlier on? So I think conventional MRI will still have a role, but we do hope that the Myelin imaging can at the very least supplement it fairly heavily. I'm answering all the questions. Sorry.
You did great.
Yeah.
Great, thank you. Now moving on to your micro MRI, your portable scanner. How long does it take one to capture an image on that scanner, and what are the qualifications? Are they as rigorous as the typical MRI?
Great. So I'll maybe throw this to Adam just to spread it around a little bit.
Yeah, for sure. So I think the company, when they designed it, they were being pretty clever, because they realized that people, even if it was way cheaper, they're not really happy for scans to take 4 times as long or anything. So what they actually did was they targeted having pretty much the same exam times. So for example, a bunch of those structural images that I showed in my section of the talk, those would take on the order of around 5 minutes kind of thing. And the image of a brain that Sharada showed, which was actually my brain, I'm quite proud of it, that was acquired on this scanner right here in just a little over 5 minutes. So long story short, the scan times are actually very, very similar. So the main difference is resolution. So there are sort of like 2 main factors with the images, which is like contrast. So that's kind of like the shading of the image, and that's important for seeing things like lesions. And then the other is resolution, which is just how tiny your little 3D pixels are. The contrast is very comparable. It's as Sharada showed, it can really pick up the things that we need to be able to see. And the resolution is a little bit lower. So the images look a little bit fuzzier. But for most pathology, there are actually relatively few cases where you need super, super high resolution. So the way we see it is like the big expensive scanner is kind of like a Swiss Army knife that can do anything. It's got a million tools. And this is just like a hammer. It's what you need like half the time, and it's just as effective. And so, yeah, as Shannon hinted at, it'll be interesting to see how that affects the way people move through the healthcare system.
Yeah. In terms of running it, it runs off of an iPad, and we were running it within about 5 minutes after getting it out of the box when it arrived. So it takes very, very little training. It's pretty much plug and play. So you pull it up and you say, I suspect migraine, and you pull up the migraine playlist and you hit go. So it's a little bit ridiculous.
Kids can run it.
Yeah.
Like anyone can run it.
Yeah.
Really, so.
One of our friends had his 5-year-olds run it on his 2-year-old sister. So yeah, it's yeah, and it's completely safe. So there's no harmful effects. So the only dangers with MRI with the higher magnetic field is with projectiles, because the magnetic field strength is so high in our normal clinical scanners, and something can go flying into the scanner. That's the primary risk. Or if you have metal in your body, it can heat up. And those risks are much lower with the scanner. So we do obviously still try to keep taking all the same precautions so we don't lose all of our good habits from when we play on the big scanner. But there's no known harmful effects to getting repeated MRI.
Great. Yeah, that was another one of the questions actually, was if MRIs are harmful to your body. So thank you for touching on that. And then I'm not quite familiar with this work, but is this linked to Dr. Basser's work on water migration on probe brain structure?
Yeah, great question. So that's Peter Basser's work on diffusion imaging. So yeah, diffusion imaging is another flavor. So one of the really cool things about MRI is that by changing the settings, as Adam was talking about, and programming how we manipulate the water and the signal from it, we get completely different contrasts. And so one of the things that we can look at is the movement of the water. And so it's in a hand wavy way, you can take a picture and then wait a very short amount of time and see how the water's moved. And if it is in a myelinated axon, so if it's in one of these wires, it's going to tend to move along the wire rather than across it. And so we can look for that. Is the water more likely to move in one direction than in any direction? And so you can use that to start a map where the different pathways in the brain go, which is a super cool and useful technique. And it is related. The difference being that what we're doing with Myelin water imaging is specific to myelin, whereas that diffusion imaging will tell you, like, if you have a structure that is just very ordered. So if you have like a whole bunch of axons, even if they've demyelinated, you'll still see that the water is tending to travel in that direction. So it gives us more specificity. Actually, Sharada's PhD project is going to involve kind of pairing these things up so we can get more information about the myelin and the axon health all together. Do you want to say more, or did I just pretty much cover it?
Yeah.
So great question.
Wonderful. Thank you. And we're just about to wrap up questions here. I see one more that we can ask. Is that the portable MRI scanner, are you also able to do the Myelin water imaging on that as well?
Great question. So we're working on that right now. So yes, ideally we'd love to do that. It probably won't be exactly the same way that we do it on the bigger scanner, but some of the techniques that Adam developed that make it so much faster on the big scanner can apply to doing it maybe not quite as fast, but with less signal that we get in the smaller scanner. And we're also playing with some other techniques that are sensitive to myelin as well. And then we can kind of compare that to our gold standard Myelin water imaging on the big scanner to see how much they pair up and where they might differ, just so that we can interpret things properly. But ideally, yeah, hopefully.
That is very exciting. And one last one here. Is there any research on drugs that build myelin?
Yeah, so it's really exciting. There's at least 3 different pharmaceutical companies that I've talked to in the last couple of years that have drugs within usually around phase 1. So there's typically 3 phases to a clinical trial. And so phase 1 is just the very first safety testing. And they're just starting to get to that they think is promising that they can build myelin. And so the question is, how do they put that into a clinical trial to prove that it's doing what they say it's doing without hurting anybody? And so this is why this is a really important thing to be developing right now, and is very exciting for us to be able to be part of that journey. So yeah, fingers crossed, there should be things coming forward that at least help or support remyelination after injury. And then outside of drugs, Poljanka also touched on some of the other interesting therapies that are coming about, like this one for the spinal cord injury, where you have people walk really fast downhill and it's stimulating repair in some way that I won't try to explain because I'm not a biologist. But so there might be other interventions outside of drug or in addition to drug that can be helping to solve this. And we just need a way to measure it so that people keep doing it.
Awesome. Wonderful. Well, thank you all so much. That was really fascinating. And thank you for answering all of our questions. I think that's all the time we have for today. So that will conclude our research seminar series. A huge thank you to all of our wonderful speakers, Dr. Shannon Kolind, Adam, Poljanka, and Sharada, for helping us learn more about new imaging techniques and the opportunity to participate in some of your studies. Thank you to everyone for joining. Our next research seminar series will be in May, and we hope to see you there. Thank you all, and enjoy the rest of your afternoon.
Thanks for having us. Thanks for all the questions.
Thank you.
Thanks.
Go ahead.